Welcome, everyone. Thanks for joining today's web meeting of the NIFA-funded project, A Systems Approach to Securing the New England Milk Supply in the Event of a Foot and Mouth Disease Outbreak. This is June 27th, 2024, and I'm really excited that we have two presenters with us today from USDA. We have Dr. Christy Loiacono, who is the coordinator of the NAHLN network, and she'll explain a bit about what that is. And we have Marta Remmenga, who's a statistical consultant. Am I getting that right? She's got all the statistical background that we need to answer questions that you may have about, well, what do we know about the bulk milk test for foot and mouth disease and how it might best be applied if we have different needs in the event of a foot and mouth disease outbreak. But this is great. Great to see everybody on the meeting today. And I should introduce myself. So I'm Julie Smith. I am in the Department of Animal and Veterinary Sciences at the University of Vermont. I've been working in the area of biosecurity specific for livestock disease prevention for a couple decades now. And I got hooked into foot and mouth disease preparedness about 20 years ago and have really taken an interest in how to bring industry needs into the picture of planning for a good, solid response in the event. And we hope we never get there, but in the event that we have something like foot and mouth disease. Interestingly, as I think all of you know, we are dealing with an emerging disease event, a new encounter with H5N1, which normally stays in avian species, but this particular virus of avian influenza has affected many, many species around the world and has made its way into dairy cattle. And so some of the things that we're talking about today have some parallels in discussions that are happening right now related to the outbreak of H5N1 avian influenza in dairy cattle. With that, I'm going to turn to our presenters and let them both introduce themselves before we get into the discussion. So Christy, if you want to go ahead first.
Thanks, Julie. Hi, I'm Christina Loiacono. Good morning, everyone. I am with USDA APHIS and currently the coordinator of the National Animal Health Laboratory Network. I'm a veterinary pathologist by training and did get to work with the National Veterinary Services Laboratories as a pathologist in my early career. And now am coordinating the activities of about 63 state and university associated animal health laboratories that make up the NAHLN and do surveillance and outbreak response testing for the nation. So I'll stop with that.
Hi, everyone. I'm Marta Remmenga. My background is in statistics. I got my training at Kansas State University. I was a professor for quite a while, 15 years, and I've been at USDA APHIS for quite a period of time now. I've worked in the area of surveillance ever since I joined. We have at USDA APHIS within veterinary services a group called the Center for Epidemiology and Animal Health that contains a lot of veterinary epidemiologists, economists, statisticians, and within that we have a subgroup that is focused on surveillance, and I've been with that group ever since I joined USDA APHIS. So my job has been to really apply my statistical trade to the ideas of surveillance and have had a fair number of years of experience in that area. I will say I started my young life thinking I wanted to be a veterinarian until I passed out when an animal was bleeding. So I went to a computer, and I'm happy to be on a computer ever since.
I'm glad to have you here. And Marta, before we turn it back to Christy, can you tell us in a nutshell what is involved in surveillance from the USDA perspective? Because I think some folks may not know what you mean by surveillance.
Sure. So, some people think of surveillance as maybe just sort of encapsulating a lot of response activities. It's really specific to gathering information. So I think of surveillance like, imagine that you're writing a research paper and you need to gather some information to address a hypothesis of some sort. Surveillance is nothing more than gathering that information to answer some questions about a hypothesis. I think one thing that's very different, we use the term monitoring and surveillance a little bit different in the United States, because specifically in USDA APHIS, when we are talking about monitoring things, those are often covered by our National Animal Health Monitoring System, which is covered by the Statistical Protection Act. So it's private information is collected. So if you've ever participated or been aware of a NOM study, that information is protected. In my group, we deal with surveillance, which is actionable. And so because we are a federal regulatory agency, in our agency, the data that we collect under the guise of surveillance is going to potentially drive an action. We don't always act on it at a federal level, but we have the authority to do that when we're conducting surveillance. I use that term a little more broadly because I think you all, with producers and the people that you work with, are gathering data and information according to statistical approaches and scientific approaches that are appropriate for making decisions, but they're not under a federal regulatory action. It is your own monitoring, if you want to say, or your own data collection. So when I say surveillance, I mean the study design that you conduct to answer a specific question. Does that help, Julie?
Yes, thanks for defining what you mean by surveillance and the distinction between surveillance and monitoring by USDA. That's very helpful. So we're going to go ahead and dive in, and Chrissy's going to lead off, and we want to start with just a background on what kind of tests are available for foot and mouth disease, and maybe help us better understand how this PCR test works.
Great. Thanks, Julie. And really do appreciate the opportunity to talk to you about foot and mouth disease today, and specifically about testing of milk, bulk tank milk samples. FMD has been part of NAHLN testing since pretty much since the beginning of NAHLN, which has been over 20 years ago. As I mentioned, we've got 63 laboratories currently within the NAHLN, and about 48 of those are approved to test for foot and mouth disease, meaning that they have analysts that are trained and proficiency tested to do the PCR testing for us. And if you look at the handout, you'll see a map of those laboratories and the distribution of them across the country. This is, again, a very timely meeting, as we've been talking about testing milk samples for FMD for years. I'll be honest, it was evaluated probably 2010-ish or around there as a potential sample for consideration or an approval to test for FMD. It went through a process of being evaluated and with our validated assay, PCR assay, and was deemed to be a good sample, but not one that we would test for in routine testing for foot and mouth disease. But it would be something we could have in the toolkit should we have an outbreak. So that the intent for years has been that milk is a good sample for FMD, not the best, but a sample that we could use, but we would never use it unless we got into an outbreak situation. So that's where we've been for, again, years. And then this meeting was set up, and I was asked to present, and I think it was, it was great. And then HPAI in cattle happened. And all of a sudden, we are looking at the milk sample using a PCR assay. So we're learning a lot about the use of milk as a sample for testing through the PCR assay. A lot of the lessons learned, I think, will be appropriate as we move forward and consider the use of the sample for FMD testing. But I will say that the virus is different. The influenza virus is much different than the FMD virus. It affects animals very differently. Influenza affects animals very differently than FMD. It shows up differently. It causes different clinical signs, and the samples have to be processed and handled differently based on how the virus affects the animals. So we can circle back to that, but let me start by talking about foot and mouth disease. virus. So this is a virus that's highly contagious. It's one of the most contagious viral infections on the planet, to be perfectly truthful. It affects livestock and includes cattle, swine, sheep, goats, and other cloven-hoofed animals, including wildlife ruminants. It does not affect humans, which is, again, a good thing. Just to parallel with influenza, it's a very different beast because it is a zoonotic. It has zoonotic potential. But for foot and mouth disease, it's very important to understand that this virus does not affect humans. There will be a there will not be a high mortality in adult animals with FND. However, there's huge economic losses. In the younger animals, we can see high mortality based on the effect of the virus on the heart of these animals, plus the potential that their dams can't allow them to nurse because it's just too painful for them. The virus is an aflovirus in the family Polymerase. There are 7. This is interesting too. There are 7 different serotypes, each of which has an endemic region worldwide, different areas of the world. Each strain requires a separate vaccine to provide immunity, and there is very little cross-reactivity. So when an infection occurs, we have to know which serotype we're dealing with in order to utilize a vaccine if that's the plan. And then again, it does not provide cross-reactivity, very little, if any, to other serotypes. The virus is found in all excretions and secretions. This was something that was driven home to us in veterinary school. This is a virus that comes out of any activity of the animal: breathing, milk, feces, urine, any excretion or secretion. They can aerosolize the virus and infect animals just simply by breathing. Pigs are our amplifier. They certainly produce quite a bit of virus and can spread it significantly. Cattle show us the clinical signs. Sheep and goats are somewhat of our kind of in the back room. They can produce lesions, but not quite as much as cattle. They can shed the virus, but not quite as much as pigs, but they do play their role. The virus can be present in milk and semen for up to 4 days before animals show clinical signs. So again, milk is a sample that can provide some good diagnostic information. May not have the most amount of virus, but could provide, again, an early detection. The virus can spread from animal to animal and from the environment to animals. So people can carry it, vehicles, feed, meat, bedding, any of these fomites can carry the virus from farm to farm, from animal to animal. And animals that recover from infection can sometimes carry the virus and initiate new infection. So they would be they could have a carrier state. Typically, the clinical signs occur as what we call vesicles that appear to be blisters. And they can be seen on the nose, inside the mouth, on the skin around the hooves, and even on the teats. And this is where animals, although it doesn't kill them, they have trouble walking, they have trouble eating, they have trouble nursing their young. The ruptured blisters can also result in secondary bacterial infections. So again, mortality is very low. However, it's always possible that a secondary bacterial infection can put an animal over the edge. Those blisters, once they've ruptured, can heal in about a week to 10 days. And animals can come back to production, but again, the economic loss while they're unable to walk, eat, nurse is significant. And at the same time, they're spreading the virus to others. Other clinical signs that might be seen are much less specific. Fever, depression, hypersalivation, anorexia, weight loss, milk loss. Those are a lot of the early clinical signs that might indicate something's wrong. But if that producer is not looking very closely at the animals, they might miss an early vesicle or blister. Now, because FMD is not present in this country, and the clinical signs, if the animals are being observed closely, are relatively easy to detect, we do rely on passive surveillance to detect it, meaning that we test based on seeing suspect clinical signs. We don't have a routine surveillance because that cost-benefit ratio, it would cost quite a bit to test enough cattle to be able to significantly say the disease is not present versus waiting to see, okay, we've got an animal with obvious clinical signs, we need to test that animal. There are a few other diseases that have similar suspicious clinical signs, but they're also very uncommon. So malignant catarrhal fever, vesicular stomatitis virus, bovine papillary stomatitis, some of those can cause lesions that are suspect of FMD, and those would be tested through a foreign animal disease investigation. But again, to date, we do not have this virus in the country. So on the handout, again, you can see evidence of infection, and the pictures that I've got there are from the Plum Island Animal Disease Center. So these are actually infected animals that have been evaluated and the pictures are from them. Okay. So what is the official testing for foot and mouth disease? So this is completed only at NAHLN laboratories. So there may be other laboratories in the country that offer foot and mouth disease, but if they are not within the National Animal Health Laboratory Network, their testing is unofficial and unsupported by USDA. As I pointed out, we have the 48 or so laboratories around the country that do the testing for us. The samples that are approved at the NAHLN laboratories include the vesicular fluid, so the fluid within the blisters, and that can be drawn out with a syringe, or if the blister is ruptured, a swab of that material can be submitted. And then the tissue around that ruptured vesicle, that epithelial tissue, can also be collected and submitted for testing. I do have milk on the list of approved sample types. As I mentioned earlier, it has been evaluated with our validated assay, but again, it is considered only relevant in an outbreak situation and not part of any approved testing now. So the sample can be tested at NBSL at the reference laboratory, but is not approved for testing in a NAHLN laboratory. The approved assay in the NAHLN laboratories is the polymerase chain reaction, or PCR, and this identifies the presence of viral particles. The PCR targets a specific region in that viral genome, and then it provides basically a molecular photocopying. And if you look at the bottom of the handout, you'll see kind of just a little cartoon of how it how it might work. So we photocopy 40 times. So that's 40 cycles. And we identify each cycle could become a threshold. So we've got cyclic threshold, or the CT value. If there is a lot of virus present in the sample, it won't take very many cycles to reach that threshold. So you might, in 15 cycles, identify enough virus to be measurable. If there is less virus in the sample, it might take 25 or 30 cycles of copying in order to get enough virus to measure. So if a CT value is low, meaning it doesn't take very many cycles to find it, then there's a lot of virus there. If the CT value is higher, it takes more cycles to copy more virus to get to that threshold. It's very important to understand that the assay tells us that viral particles are present in the sample. It does not, however, indicate that the virus is infectious. So in order to determine if what we find in a sample, if those viral particles represent a virus that is alive and potentially capable of replicating and shedding and infecting other animals, we have to try to grow it in cell culture. So that is a step that happens at NVSL. The non-laboratories do our screening tests with PCR. They identify the viral particles are present in a sample. That sample is then transferred to the National Veterinary Services Laboratory, or NVSL, for confirmation. So they may redo the PCR, or they may go ahead and just put the virus on cells and try to grow it to identify whether it's infectious. They will also sequence the virus. Right now, that's not being done in non-laboratories. The only test being done in non-laboratories is PCR, but NVSL for confirmatory testing would include PCR, virus isolation, sequencing, and they may also take serum and do serology looking for antibodies. So next question, how do we choose an assay for use in official testing in the non-laboratories? So as I pointed out, we do have the PCR that has been approved. It's gone through a validation process so that the PCR itself is validated. Then we have to look at the sample types that can be approved. And the ones that I've mentioned already have been evaluated using the validated assay and either approved for the NAHLN testing, which again is vesicular fluid, swabbing of the vesicular, of the ruptured vesicle, or the tissue around the outside of the vesicle. That's all at the non-laboratories. At NVSL, they can also add samples such as milk, urine, feces. They can do any sample that would be collected for confirmatory testing, but not necessary to confirm a case. For assays that are deployed to the NAHLN, the first requirement is that they are either produced by NVSL, by the reference laboratory, and validated, or they're a licensed kit in the United States. And that licensing happens through the Center of Veterinary Biologics. They do a form of validation. Once a kit is licensed in the US, then for non-approval, we actually do a second level of evaluation because licensure doesn't necessarily provide information on whether the assay will be useful for our purposes. Bottom line, our purposes, whether the surveillance that we're doing or the use case that we need to address can be provided through that assay. So NAHLN will do further validation, evaluation of a kit once it's licensed in the United States for our purposes. And the validation process is we follow one that is has been published through the World Organization for Animal Health, formerly known as OIE. So there's an OIE chapter on validation, and Dr. Remmenga will provide more of the specifics around what the validation process looks like. But for non-purposes, two levels, either NVSL developed and validated or licensed in the United States, and then further evaluated for use and approval for NAHLN, and then deployed to the non-laboratories.
So before we get Christy off the hot seat, I've got one more question about testing and sample types. And so you've been referring to a milk test PCR, and I'm wondering, when we talk about a bulk milk test and using the PCR in bulk milk, is that a different ballgame? And what do we need to know? And maybe that's the handoff question.
Thanks for that. Yes. So one cow producing milk it's a very easy one-to-one evaluation to make. Even pooling a certain number, say 5 cows, it's pretty straightforward to understand, can the dilution factor of putting one infected cow in with 4 other cows' milks affect the ability or the sensitivity of the test to detect virus? Relatively straightforward. When we're talking about bulk tank milk, we don't know unless we're told, we don't know how many cows submitted milk to that sample, how many of those could be infected at what level. Something that we're finding out with HPAI is if some cows have antibodies, if they're developing antibodies, how does that affect the ability to detect what virus is present in that sample? So there are many more variables that have to be identified and evaluated. And I do think this is a good spot maybe to hand it off to Marta to talk more about how that evaluation could happen. Over.
Is that my cue to come on?
If you're willing.
Yes, I am. Hello again. I just want to reiterate that I am not a veterinarian. The sight of blood caused me to pass out when I was 18, and I moved on to computers and statistics. I have been working in the area of epidemiology, animal health epidemiology, for a number of years. So I sometimes say words and pretend that I know things. So if I say something that is actually technically very wrong, just correct me, and I do not mind being corrected at all. I'm always learning. And so one of the ideas I want to get across to you is that when we are doing statistics, one of the things that's very, very important is that we have an accurate measurement of whatever it is we're trying to measure, and we have a clear idea in our mind of what it is we are trying to gather information about. I don't think anybody here that's done a research project would just go out and say, I've got this great tool, let's just go use it, and then decide what research questions we want to answer later. And so that's one of the things you want to keep in mind as you are using this fantastic tool of a RT-PCR in bulk tank milk is, what are you trying to answer, and is that tool appropriate? And I've told this particular story before, and hopefully it'll make sense to you. We have big truck scales out there and large animal or scales that are like truck scales that you might use just to sort your cattle into small, medium, and large pens. If you were doing a research project to maybe improve genetically the weight of your animals, you would never use a truck scale to try to determine whether or not your genetics were producing great differences in your animals. You would use a much more precise tool. And so you want to use the tool that goes with the questions you're answering. And for things like bulk tank milk and other aggregate samples where we are collecting a lot of information about a lot of animals at the same time, there's a tendency to say, this is great, let's use it for everything. And that's like saying, this truck scale is great, I can just walk my cow up there and weigh them. I don't have to do any precise anything or calibrate it. We just walk them on there. And then say, well, then let's use that for everything. And I think, everybody would recognize that's not a good plan. And so I want to talk about a couple of things that are specific to diagnostic testing, which are a couple of characteristics called sensitivity and specificity. And those are typically defined at the animal level. The diagnostic specificity for a test method, and I'll say this, and I think it's in the PowerPoint. I don't know if Joanne is going to hand that out to you, but she's got some technical details in there that you can read later. But the diagnostic specificity, you take a group of known uninfected animals, which we have, imagine you've got uninfected animals, which we have hundreds and thousands of those in the United States, right? So we can test for the specificity of this PCR. We can take animals that are uninfected and look to see whether or not any of them ever show up positive. Those would be false positives. So specificity is the proportion of negative animals that you get out of that group of uninfected animals. And it is high, 99.99 something. Who knows? It's very, very high for the bulk tank test. Sometimes tests will trigger positive because of something the cow ate. You just never know what might make a false positive until you've evaluated that test and explored it. And I suspect, having never been in the lab, that I suspect that there are lots of test procedures and assays that people have tried. And it's like, that doesn't work, because as soon as the cow eats clover, it triggers the test. And so we can't do that because lots of cows eat clover. I'm making things up, but you understand that there is a whole process to getting to that point where we know this test is very specific. Tests are often so specific that you've got to have the diagnostic test assay set up so that it picks up the strain. So it might not, if we use a test, and we know that with human flu, if we've got a particular flu, test might not pick it up until they tweak that and get it set up for the right strain. And some tests are very, they're not that specific to strains, they're just specific to the virus. So we kind of need to be careful that we use the right tool for the right situation. So that's diagnostic specificity. It's very specific to the virus that you're trying to pick up. The other one's diagnostic sensitivity. And Dr. Loiacono has already pointed out the importance of not having false positives. That can trigger something that is really detrimental at an economic level, a trade level. So there is a lot of procedures and processes and protocols to make sure that we do not get it out in the newspaper that there's a positive, only to find out there isn't, because retracting that can be very, very difficult. So we work really hard to be very positive, very sure about our positives. The flip side of that is the diagnostic sensitivity. So there we're talking about the proportion of samples from known infected animals that test positive. And we can have less than perfect sensitivity. We can walk those animals onto that scale, and for some reason they don't get weighed at all. Their weight is just totally off. It just, there's something wrong with the scale that day, or there's something wrong with what's going on. So this test can be less than perfect, and it can fail to detect positive animals. One of the ways that can happen is we chose the wrong sample type. And so maybe milk is a great sample for one virus, and it's not a great sample for another virus because that virus is not secreted into milk. And so in this particular case, FMD seems to be coming out of every orifice in the animal's body. It looks like it's a good sample type, but that's not always true. So you can't just assume milk is going to be a good sample type for every virus. There are periods of time during the infectious stage of an animal where any given fluid might not have much virus in it. So you might be too early to pick up virus in urine, or you might be too late to pick it up in blood because they've moved out of that having much virus in their blood, and it's all antibodies. So you have to also recognize that there's a time period that is important. On an individual animal, that sensitivity is typically defined, and it is set up with infected animals, which those studies have to be done in another country because we do not have FMD here, or they need to be done in a highly biosecure laboratory that is kind of an artificial situation. But that's what we have to do to be able to get an infected population and determine the sensitivity of the test. So now you move to bulk tank milk, and now we have not only the way the virus moves through an animal where they have a low response to it, they have some virus, then the virus gets very heavy, and that's, if I have the flu, that's when I might be really, really sick, and then you start to recover and it goes away. But now we have multiple animals, so you kind of have to imagine the spaghetti string of animals coming in at different phases, and you have, as Dr. Loiacono pointed out, some animals that are recovering and have antibodies, and they're putting antibodies in the tank at the same time as another animal is just barely getting sick and only putting a little virus in. And so we've got this whole phase now that is spread out longer than an individual animal. And we have to think about what is the sensitivity of a bulk tank milk test on a group of animals. And now it's not just one size fits all. It's not just there is one sensitivity. There's a sensitivity early on. There's a sensitivity that's late. Right in the middle of the outbreak, when there's lots and lots of infected animals, it's likely a very sensitive test because if you have enough cows with CT values that are low enough, we don't have to worry about the dilution. Early on, there might be a period of time where we would miss that disease. And so this then ends up helping us think about how we might want to use that test. The other thing that happens, and in your handout, there's a diagram that's sort of a very generic setup there in the PowerPoint. If you've got that open, It shows you a couple of curves and a cutoff in between, and that's sort of the same thing on this first diagram that is in your presentation with the orange and green box. There's a cutoff in there, and that cutoff is part of the validation process. For the PCR for FMD, I hope I have this right, the cutoff is typically 40 cycles between distinguishing whether or not an animal is infected or not. I have been aware of other tests where they've adjusted that cutoff, but that's part of the validation process. That doesn't happen later. It happens very carefully as part of the laboratory process. But you can see if I move that cutoff on that orange and green box, I could get my sensitivity so that it's perfect. So I move it clear up to 50 on the orange and green boxes, and I would have a test that's near perfect in terms of sensitivity, but I've just blown my specificity right out of the water. I probably have something like it looks like 60% of my cases would be false positives. And so there's this really careful balance between what that assay does in animals who don't have the disease versus how it behaves in animals that do have the disease. And that is part of the very sensitive process of doing validation. What typically happens in the diagnostic laboratory is we aren't going to shift that. Once that's sort of been balanced for an individual animal, we aren't going to shift that for bulk tanks or for aggregate samples. We would end up chasing our tails because you get one tank where you could separate them really great because you happen to catch the infection at a really hot time. And then the next time you turned around, you would have a terrible test because that particular cutoff wouldn't work. So it cannot be a moving target. It's part of the validation process. At the same time, identifying exactly what proportion of your units can be identified as truly positive from the test and what proportion of uninfected units can be determined as truly negative from your test is part of the validation process. So we have to do that for bulk tank, but it is a very, very difficult process because that means looking at lots of different kinds of combinations of infections of cows and different dilution levels and different stages of disease. So it is something that we hope to do as we learn more about the test, and we hope in some ways never having to do, because we hope we never have FMD. But if you look at that second diagram, I've tried to present something that might be more realistic of an aggregate sample. Problem is where that orange box flows and how spread out that is a function of where we are in the disease and how many animals we have in that tank. So if I just have one or two infected animals that are with CT values themselves in the 35 to 40 range, and I dilute it with thousands of other cows, I might be way up there in the 60s and 70s on that tank. You just would not pick it up. A few animals, some 15s and 16s, maybe amongst 1,000 cows, could be down there in the 30 range. So we don't really know what that orange box looks like for every bulk tank. It could be different in every stage. So we have to be really, really careful what we do with the bulk tank test in terms of use cases. So to kind of drift into that sort of thing of talking about use cases, Dr. Loiacono has already pointed out how the PCR cannot tell us if the virus is infectious or not. So a very simple, I hope obvious use case that we would not want to use the PCR for bulk tank test is to tell us whether or not our cheese is safe. That just doesn't work for us. We're taking the bulk tank milk, which is raw at this point, hasn't even been through pasteurization. Perhaps we've identified the viral particles in there, but that's not going to tell us whether or not the cheese that's coming out of the processing plant is safe or whether the milk on our shelves is safe. The RT-PCR is not the tool for doing that. Dr. Loiacono has talked about the other diagnostic tests that would be used in that situation if that was your purpose. So I'm going to move into a little bit more of a surveillance way of thinking about the world. Remember that with surveillance, you're trying to talk about what actions what questions you're trying to ask and what you're trying to answer with that particular test. For at a federal level and in a regulatory setting, or even at maybe at a state level, and maybe even for you as producers, there might be a variety of goals. You might be looking to see whether or not you can support animal movement. That might be one goal. Another goal might be that you want to just simply demonstrate that the control actions we've used are working. Those are two very, very different goals and require a different level of surveillance. And remember that I'm going to get kind of a little bit picky about language here. When we talk about goals, those are kind of vague and they're not necessarily measurable. When we start talking about objectives, we're looking at something that is more specific and measurable. So I might have surveillance objective that is set up to identify or detect an infected animal when the level of the infection is 2% or more. So that would be a very measurable objective. Another one might be to detect if an infection is occurring in herds at the US level, if infection is at a 10% level among herds. That's a different kind of an objective. It's measurable, but it's at a national level and a herd level. Setting the objective that we need to meet a particular goal, like animal movement or control spread, we need to be aware of the special conditions that are occurring at that time in combination with the tools that we're using to make that surveillance and the specific surveillance objective we've set. So the combination of your surveillance goal and that context of what's going on at the time might be the use case. So let me give you an example. Suppose we're collecting information on a single farm. We know it's had contact with an infected farm. So it's one of those that the state vets traced out. If that farm is located in the control area, then it's going to be subjected to a variety of quarantines and animal movement restrictions. And that's completely different than if that farm that we traced out is outside the control area and has no movement restrictions on it. So if our goal is just to determine whether or not that farm is infected, to kind of figure out the extent of the outbreak and get a handle, get our heads around where the outbreak is, if the farm's in the control area, I might be willing to test at a 10% design prevalence level, knowing we're going to be back next week and they're not moving any product. That farm that's sitting outside that control area that can move product tomorrow, I'm probably not going to be happy with a 10% surveillance objective. I may want something very low, and I may even want to do something that requires repeated testing through time and have some sort of a mini restriction on the movement of that farm because it's been traced out in combination with my testing. Because the minute we decide that farm is not infected, it may end up, depending on how the state or other regulatory people decide how to handle those contact farms, it's gonna maybe be able to move product fairly quickly. So we may want to be a lot more stringent on that farm that is outside the control area. So that's what I mean by use case. It's, we're trying to detect whether disease is on the farm, but the within control area and outside control area forms two different use cases because of the context that's going on there. The other thing to remember about statistics is, and these sort of studies of surveillance is, we are not going out there and looking at every single animal. Even with the bulk tank test where you think you're getting all of the cows, there are cows on that farm that have been removed from the bulk tank. So it's almost reverse targeting. We would love to, in most diseases, say, give us your sick and dead and let us test those. Well, the sick and not doing wells are pulled out of that bulk tank. And so now they're in a sick pen somewhere. If there is a way to get at that milk, we would probably really encourage that, and it would be a good thing to do. It's obviously not as convenient as the bulk tank because they're already collecting a variety of milk samples for other testing. That's very handy. And we're not doing that with the sick tank because we know those animals are sick and we're not testing them for white blood cells. We know they're kind of there. So we're trying to gather information when we do statistics about the whole herd, but we can't look at every animal. So we're choosing a subset. And in this case, we're not going in and randomly selecting animals. We're selecting the animals that have been put into that bulk tank, which are your healthiest animals. So we kind of need to look at what's happening there and design our surveillance in a way that's examining those consequences of the errors. So if we've pulled the sick animals out, we've got mostly a healthy tank. It's also very, very dilute. And we missed FMD because it was early in the stages. There weren't vesicles. There was some fever or some doing poor. And off the milk goes and off animals move. We've made a horrible, horrible mistake. And it is not the same sort of mistake as telling the world we have FMD when we don't. It's the other kind of mistake that we have now allowed disease to spread when we shouldn't have. And so the consequences of that are a different kind of consequence, and they have their own sort of problem with it that we have to be really aware of. At the same time, and I always joke with my colleagues, because as a statistician, I'm always going to ask you for way more sampling and way more data than you ever want to give me, and probably sometimes more than is physically possible to get. And so we have to balance that. So, would it be my preference that we go around and get some sort of a milk sample or a oral swab or a probang sample from every cow on your farm? Absolutely, I would love that, because now I know what's going on at that farm as best as I can. That's ridiculous. It is not feasible. And so that balance between the consequences of making the mistake when you have a less than perfect test and the practicality of what I would prefer we would do if I wanted to be perfect has to be sort of balanced. So you want to take the important actions to reduce the negative consequences that can occur from those erroneous decisions. And sometimes that means not relying on only surveillance. Conducting surveillance, getting negative results, and then having something that says, and oops, if we are happening to move animals that are infected, we've got a quarantine place, we're holding them on the other side before we just mix them in. And again, I know I'm asking for things that may not be physically feasible, but you get the idea that, we may say we're moving animals to slaughter. That doesn't mean you don't wash the trucks. You don't do all of this other activities that could mitigate if we happen to have some cows that are infected moving on that truckload out. Just because the test is negative doesn't mean we've completely wiped the slate clean and we can behave as if there is no virus there. So you really want to have those other mitigations in place. So let me just go back through. We're going to conduct surveillance on let's say we're conducting surveillance on an infected herd and we failed to detect it. That could happen for an individual animal simply because we're testing a subset of animals and we did not collect a specimen from any infected animals. That's one of the ways it happens, because that's statistics and there's sampling error. There's a couple of ways to decrease that error. One of them is to sample a larger number of animals. That way you're less likely to miss the subset. And the other thing is that well, I said two things, but that's typically what we do with individual animal sampling, is you collect more. We have a hard time with that with a bulk tank, right? If we go in and get a bulk tank and that's all of the animals, how do I get more bulk tank? If I just test the same tank again, that's not getting me that sampling error issue. The issue with the bulk tank may simply be that the virus is just very low because there aren't very many infected animals. Testing that again isn't really going to help me. So one of the ways we do that with bulk tank is we say, we're going to hold things and we're going to come back tomorrow. That does two things for us. That's where my 2 comes in. One of them is it gets me a second look at that tank in case it is something that happened at the lab or part of the assay that caused the failure. Other thing it does for us is that the virus is going to spread, and so the concentration may go up. And so it gives us a little bit better chance at picking that up in the tank. We also have just the whole concept of, if we're moving animals, a pre-movement isolation period where we say, please just don't let anybody on and off your farm for a little bit. You've tested once, let's test again before you move product. And that way we can be sure that if it were there, it would have spread enough that we could have detected it. So I hope you can see that using bulk tank milk is a really cool tool because it would get so many animals at once. We are also focusing on the more healthy group, which makes it a little bit harder. We've got the potential for dilution, which means depending on where you are in that spaghetti diagram of different animals in different stages, you might miss it in that bulk tank. So we want to be careful about recognizing that it's a less than perfect sensitivity. And I think depending on whether you're pulling it from a silo that's really huge and has lots of tanks, lots of milk in it, and you're thinking about using it where you've got lots of farms in the same tank versus a smaller tank where you've maybe got 500 animals in it, that's going to change that sensitivity of the test. And then, of course, if a farm has multiple bulk tank, you start thinking, well, if I just did one bulk tank, you know, what if I did all of them or some subset of them? Now you have sampling at the tank level. So you could miss the tank that has the infected animals in it. If you did them all, that would be a cool thing. As far as a statistician, we love censuses. We love it if you can do that sort of approach. So a number of years ago, we evaluated the bulk tank test and picked up some ideas about where we thought that test was appropriate for FMD. And that document is available on the web. You can look up evaluation of our RT-PCR in bulk tank milk. And it's an older document at this point now, but I think it still applies. And I think as we learn more with the H5N1 infection in dairy cattle, we may want to revisit and reevaluate the bulk tank test for FMD. But at the moment, one of the use cases that's just not appropriate is to talk about safety of products that are going to be pasteurized. The PCR will pick up that there are viral particles in milk on the shelf. That does not mean it's not safe. That doesn't mean it is safe. It's just the wrong test for deciding that. We just shouldn't be using that tool. Some other places where we think that it's not a good idea to use it, it would be a national FMD testing of all bulk tank tests, bulk tank milk samples today for FMD. And there's a couple reasons for that. One of them is it's a lot of money, and we can pick up disease with vesicles and foreign animal disease investigations would probably be a better way to do it. So that's a better tool. But the other thing is, as specific as that test is, we could, with as many bulk tanks that are out there being tested, I don't know, have 1 or 2 false positives a week. Can you imagine fighting that media, and just, you would just get worn out chasing down those false positives and trying to figure out why they happen. Why would they happen? Because there's so many tests of bulk tank milk out there. If we did them all, probability wise, it's just going to happen. It's not a perfect test. Even if it's 99.9% sensitive, 1 out of every 1,000 tests could come back positive. And there are, I don't know, thousands of bulk tank tests happening right now as we speak. In the next hour across the United States, we would be having false positives all the time. So that's a bad, that's a use that we just don't really recommend because of the problems that it could cause for us. It's also not a good thing for maintaining disease-free status because, again, we could miss it. And so it wouldn't be, we didn't recommend that for maintaining disease-free status once we establish it. We also don't recommend a bulk tank test in a foreign animal disease investigation. We felt that it was more important to go to the animals that have the vesicles that triggered the concern and concentrate on that. We wouldn't want to go to the bulk tank where you probably are not milking these animals with sore teats into that bulk tank. And, maybe there's other FMD animals in that tank, maybe not. And we would come back saying, oh, the bulk tank's negative. I guess we don't have it. We would rather have the test of the animals that are showing the signs. The other use that was recommended that was suggested but not recommended is to permit the daily movement of raw milk. One of the problems there is milk's moving so fast, we don't get PCRs back fast enough. And so the flow of commerce is a reason that a test might not be an appropriate test. Not to say that there is another test that's better. If we're worried about moving the daily movement of milk off of the farm, we're going to have to come up with some way to declare a farm as free from disease with not just bulk tank testing, but other sorts of things. And then biosecurity and, you know, just a whole array of things to be able to say, you're certified free, you can move whenever. And we're not doing it on the basis of a bulk tank test itself. So it's a bigger picture that we would be doing there. Same thing with live animals. We would not want to use bulk tank testing to say, you're free today so you can move animals tomorrow. That it's got to be a bigger picture of what's going on at the farm. The bulk tank test might be part of it, but it would not be a single, let's do the bulk tank test and then you can move animals tomorrow. Same thing with potentially contaminated materials, bedding, feed that's gone bad or whatever else. We would not want to use bulk tank testing to say it's okay to move those sort of materials. Some cases where we thought bulk tank testing with PCR made the most sense included targeted surveillance of dairy premises at the first detection of the United States or in North American countries. So suppose Mexico had FMD. We might want to set up a region that is close to Mexico, receiving animals from Mexico to do bulk tank testing as kind of a screening and sort of a forewarning. Whether we would use it for that, you know, these were just ideas in this paper. So remember, I'm not a policy creator. None of this is policy. These were ideas where we thought this might be a good use for it. As more people get involved and we think about the logistics of it, you may very well say, nope, take that one off. That logistics of that are crazy. We can't do it. It would, you know, some of these things, I have to work really closely with Dr. Loiacono and say, I'd love to do this for surveillance. She's like, whoa, you'll just kill our labs. And the labs need to be working on the outbreak that's happening. We got to come up with another way to do this sort of nationwide screening when our labs are focused on dealing with the 2 control areas that have a huge number of prems, and we're just trying to get that taken care of. So don't weigh us down with some other fancy ideas that you have in your little statistical brain. Testing of dairy premises that are not declared as infected within a control area. That might be a really beneficial just to have that ongoing evaluation of premises within a control area that says our control actions are working. We are watching them. They have not become infected. They're not moving anywhere. So our false negative rate, we're not too worried about because these are farms that have to do permit testing just to move product. But that sort of ongoing, we understand the disease has been controlled, might be a good tool for bulk tank tests. The initial information about the classification of the permanent control area might also be a good place where we come in and say, you're brand new, you're a neighbor to the infected farm. We don't know what your status is. Give us some bulk tank tests. Let's at least begin to establish your status in the control area. Again, that farm would be subject to a different surveillance for moving product. Would not be using the bulk tank to move product, but just simply to say you're an at-risk farm, you can move into the monitored status and start doing that other testing. Perhaps the testing of milk to be fed back to susceptible animals would be a good idea. But I think I did not know that pasteurization might not kill all of that. And this is the bulk tank is still raw milk. And so, you know, maybe doing the bulk tank test and saying we didn't find enough to pick it up on the PCR. Now in combination with whatever heating process we're going to do, that ought to be good enough. If you can't detect it and you heat it, it's good to feedback. Again, I'm not the person to make that decision. This was just an idea of a way that we could use it. The other place that, and we typically don't do this in an outbreak because we don't have the resources, but it might be a good tool to do kind of a flooding of sampling in the surveillance zone, especially initially as we're trying to figure out where the disease is. So we've got an infected premises. We've created a control area. The surveillance zone is the donut around there. We could probably do a quick survey of the surveillance zone and maybe pick up some places early to get some new control areas put down quickly. So that's another idea. So there's a handful of ideas where we think PCR makes the most sense. Unfortunately, it's not some of the ones we really would like let's do a bulk tank test and move our animals tomorrow. That's just not one of them on the list. So I think that sort of concludes my side of things. Hopefully I covered enough things in there to sort of give you an understanding of why the bulk tank test is, we need to be careful and we need to choose our use cases very carefully for how we use that particular measurement tool.
I want to throw a couple questions out there that came to mind when Marta was explaining the cautions about using the bulk milk test and being thoughtful about what milk is being tested and what milk is not being tested. And one thing that occurred to me is that on some farms, and it may not be true of every dairy farm, but some dairy farms, when there's a cow with abnormal milk or that's not, that they're not going to put the milk into the tank, that cow may still go through the parlor and they may take a sample or may have the opportunity to take a sample from that individual cow, even though that milk is not going into the bulk tank. And that maybe encouraging that could accelerate detection in certain situations. And the other approach would be for those in milking in parlors where they have the capacity to do string sampling so that they could pull a sample from each turn of the parlor, and whether those were submitted along with a bulk sample or stored or whatever, that there would be a way to go back and possibly test smaller groups of animals from within a herd to have a better opportunity for detection.
And I'll just say those are amazingly good ideas. And I think, again, the balance there is, I know the bulk tanks are easy because there's already milk coming out and already being sampled. Some of those others may be more difficult. The chain of custody of those may be more difficult to figure out. But I think using those and thinking through how you might get at some of those until we have a better idea of the actual relationship between our probability of detecting and dilution, stage of the disease, and all of that as FMD rolls through a farm. Until we know more about that and can say, oh, we now know, when you first get some signs, we really need the sick animals. But once you're at this point, it's going to be in the bulk tank because it's just so much all over the farm that it's going to end up in there. And we don't need that. Versus what's happening is you get a farm as it's getting over the disease and are still trying to understand what happens when there are antibodies and virus in the same milk, and the work that we need to do there. So I think anything that you can come up with like that are unique and really thinking about that process of how does the virus end up in the milk and what do we do with sick animals on the farm is really going to be a support to detecting disease on farms.
Well, the other question that I have has to do with, well, how do you get bulk milk samples from where they normally go with the milk truck to a non-lab? And I'm just wondering if that's a potential pathway. And you mentioned custody, chain of custody. Is that something that could be looked at in some of the use cases, for instance, surveillance early in an outbreak?
Absolutely. I'll jump in. We're dealing with that with the high path, the H5N1 outbreak in dairy cattle, because it varies state by state who has the authority to pull samples. And the PMO, I think, has authority in some states, the state animal health officials in other states. And as we're looking at doing the dairy herd status monitoring, which is a voluntary program to test bulk tank milk, it's on a state-by-state basis of who that state determines should collect the milk from the farms that are enrolled. So getting that addressed ahead of time, I think, would put us ahead of the game if FMD were to